Good morning, and thank you for joining us. I'm Lori Dajose. I'm a science writer with Caltech's Office of Strategic Communications and an alumna of the Institute. It's my pleasure to welcome you to the second in this new series of webinars, Conversations on COVID-19. Hosted by the Caltech Science Exchange, the series is designed to give all of us a chance to hear directly from the researchers in our community who are bringing insights and solutions to the COVID-19 pandemic. I'm joined today by Pamela Bjorkman, Caltech's David Baltimore Professor of Biology and Bioengineering. Professor Bjorkman's research centers on immune responses to pathogenic viruses, so we've called upon her expertise to help us all understand more about what's going on today with the COVID-19 pandemic. Professor Bjorkman, thanks for joining us.
Thank you.
So let's start with the basics. What is a vaccine and how do they work?
Okay, so to understand a vaccine, I guess all you need to know is that when a virus infects your cell, it then turns on host immune responses. And so what a vaccine does is it turns on similar immune responses, but is not an infectious virus. So it's something that mimics the virus enough so that your immune system recognizes it, but it doesn't cause you any sort of illness.
Yeah. So obviously things are changing quickly, but to make sure we're all on the same page, I'll give a quick recap of where we are today with the COVID-19 vaccines. The World Health Organization reports that there are 63 vaccines in clinical development, meaning they're being tested in humans, and 172 in preclinical development. In the US, 2 have received emergency use authorization, the Pfizer-BioNTech vaccine and the Moderna one. And both are mRNA vaccines, and both are more than 90% effective in large clinical trials. And there are also large-scale trials planned or in process in the US for 3 other vaccines, AstraZeneca, Janssen from Johnson & Johnson, and Novavax, all of which use methods other than the mRNA platform. So let's focus on the 2 COVID-19 vaccines approved by the FDA for emergency use, the one from Pfizer-BioNTech and the one from Moderna. So how do these vaccines work and what makes them different from each other?
Okay, so both of these are so-called mRNA vaccines. That means that what they do is deliver a genetic message to your cells that instructs them to make a part of the virus, which is the part that's normally on the outside of the virus called the spike protein. So the spike protein is on the virus, it would allow the virus to bind to a cell and then infect it. And so most of your immune response in the form of antibodies, which are circulating proteins in your blood that can go in and out of tissues and so on, those are directed towards the spike. So what these vaccines do is they deliver the genetic message for that so that the cell that takes up that message then starts making the spike and you make an immune response against the spike. So you can't, there's no other components of the virus, so it can't infect you or give you the disease COVID-19. Now, how are they different? None of us know because the mRNA is made in a standard way that researchers in biology, bioengineering, and so on, we all would know how to make that mRNA. After making it, they encapsulate it in what they call a lipid nanoparticle, which is some kind of thing that has components of membranes and cholesterol and other things in it. And that allows it to go inside cells. It might actually also stimulate your immune response, but we don't really know what's in those. And they probably differ a little bit between Moderna and Pfizer, but that's proprietary. But I suspect there's no real difference between them.
Okay, so the reason that we don't know what's in them is because that's the property of the companies, right?
Yes, and I've looked at their publications and they refer back to older papers that tell you sort of what they probably are. So we have a good idea what's in them, but we don't know exactly.
Okay, got it. So why are mRNA vaccines being used now against SARS-CoV-2 for the first time?
Well, there were mRNA vaccines that have been in humans. It's just that the clinical trials weren't completed. So, for example, Moderna had a mRNA vaccine, and I think that went into humans against two forms of influenza, I believe in 2018 or 2019. So that's been in humans. It's just that they got through the phase 1 clinical trials, but they didn't complete the phase 2 and the phase 3, which involve enrolling tens of thousands of people. And normally the timeline for doing that is quite slow, but there was an emergency in this case. So once they got the mRNA formulated into these lipid nanoparticles, they went very fast through phase 1, phase 2, and phase 3, but not skimping on any safety protocols whatsoever. It's just that they were allowed to do this at a much faster pace, and a lot of money was put into it. Because normally what you would do is these things have been tested for a long time in animal models. So the mRNA vaccines have been thoroughly tested in animals. They've gone into humans for other things. There's never been safety concerns, but normally it would take a while to complete. The phase 1 would be, is there any horrible reaction? And phase 2 would be, is there any horrible reaction and does it actually have protective immune responses? And phase 3 would be on tens of thousands of people, where you'd have enough people that you could look for rare adverse reactions. So they actually just combined, say they had a combined phase 1, phase 2, and then a combined phase 2, phase 3 to get these out faster. But there were no safety protocols that were bypassed.
Got it. So why do we have to get 2 doses of these vaccines? And do we know yet how long protection from the vaccines will last?
So this is a commonly known in immune responses, is that you mount a more strong or a more potent immune response the second time you see whatever it is that you're reacting to. And so that's what's called the boost. So in all the vaccines so far, they test the efficacy, for years in animals to see if a boost is necessary. So it's hard for me to know exactly how they came to that decision, but definitely the FDA, they put out something on January 4th, because a lot of people have been saying, let's just give one of these. We don't, maybe you could spread them out more if you just give the prime, which is the first dose. And they put out a statement saying that there aren't enough data to know if the prime would be effective, because they didn't have enough people in that arm of their clinical trial. And therefore they don't want to go against what was done in the phase 3 trials. So the short answer is boosting helps your immune system. The second or third time you see a pathogen, you mount a much stronger immune response to it because of what's called immunological memory.
Got it. So why do some of these vaccines have to stay in very cold storage while others don't?
The mRNA vaccines, the Pfizer is licensed only at this very cold minus 70 degrees centigrade temperature. The Moderna vaccine, I believe, and people should check on this, I believe is okay. It can last a month or so at a regular freezer temperature, say minus 20 degrees Celsius. The answer, someone did very careful studies of how long the mRNA actually is good and how long the lipid nanoparticles last as a function of the way it was stored. And they've come up with these recommendations for the storage. It's based on a lot of experiments. Oh, and the other vaccines are not mRNA vaccines. So they would be, well, we can get into later exactly what they are, but those components are not solely genetic material. Some of them are genetic material enclosed by some kind of protein capsid, and those are simply, they've been shown to be more stable at other temperatures.
Okay. So why do some vaccines protect you for life, whereas others need to be updated? For example, the tetanus shot every 10 years.
So I wish we had an answer to that. And I would say that nobody knows. And so the tetanus, the reason you're supposed to get a booster in the tetanus shot is that every year there's like maybe 10 cases of tetanus in the US. And when they look at who they are, they're adults who either were not ever vaccinated as a child or adults who didn't renew their childhood vaccination. So I'm sure everyone knows they got a tetanus shot as a child, and then you're supposed to renew it every 10 years. It's really rare for people to get tetanus, but when they find it, it would be someone who either was never vaccinated or hasn't renewed it. So the recommendation is once every 10 years. This is all determined empirically for each vaccine. They do a lot of tests in animal models. They look at all after the phase, they've initiated a phase 3 trial and they've completed it for both Pfizer and Moderna, but they're going to keep going for years. They're going to be studying these people. And then we will know later if these vaccines need to be readministered. We just don't have enough information right now. But for other vaccines that we all take, they've studied it for years and they've come up with recommendations for how often you should get boosted.
Got it. So we've heard a lot in the news about new strains of the virus that causes COVID-19. How quickly do coronaviruses mutate? And is it likely that the vaccines people are receiving now will protect against these mutated strains?
Okay, so the first question is how rapidly do they mutate? And I think maybe people don't realize, maybe not everyone knows that all viruses mutate because they make a huge number of progeny and they don't have complete fidelity in copying their genomes. And that's actually to their advantage because some of the rare mutations might have properties that allow them to, say, spread more. But the interesting thing is when viruses mutate, they rarely mutate to become more virulent. Usually they mutate to become less virulent because it's in the virus's advantage to infect more people and not, for example, kill them, because they'll spread more if they don't kill their host. I think the second question was, will these, the UK variant and other variants of concern, will the present vaccines work against them? I suspect they will, and the data I've seen so far are supporting this. But what I want to emphasize is that when you make an immune response to anything, that response is what's called polyclonal, which means if you're talking about your antibodies, you don't make just one antibody. So let's say here's the spike protein and here's the antibody that binds to it and prevents it from binding to your host cells. So that's the way that antibodies would stop the virus from entering, and that's the way the vaccine would be protective. If you have only one type of antibody raised, then yeah, if you mutate in the site where that antibody binds, the vaccine will no longer work. But your immune system does not do that. It makes antibodies against all these different places. And so the virus is very unlikely to mutate against the whole range of antibodies. Now, let's say, of course, the variants of concern these days are of concern because they're not just like single variants. They're variants throughout the virus, but the ones that matter for your immune response, at least the antibody part of your immune response, are the ones that change in the spike. And so it could happen that there are enough mutations that a lot of your immune response is diminished. And then the great thing about these mRNA vaccines is the estimate is they could make a new one in 6 weeks that would encode the new variant. So if that's necessary, they can do that. And it's already been in people. They know about how to formulate it. They know what safety procedures there are. They could probably get emergency use authorization quite quickly against new variants if that is necessary.
Right. Okay, so let's talk about some of the practical aspects of getting vaccinated. The initial studies on these vaccines show that they're more than 90% effective at preventing serious symptoms should a person become infected with SARS-CoV-2. How is that 90% number calculated? And do we know yet if someone can still transmit the disease even after they've received the vaccine?
Okay, so how that number is calculated is, for example, in the I can't remember now if it was the Moderna or the Pfizer, the numbers are very similar. They're like 94 point something percent. And my memory is they had 95 cases of COVID-19 disease in the whole clinical trial. And that included, say, 30,000 people or so. So they have 90 infections and 95. And of those 95, 90 were in the placebo group. So you do the math and you come out with 5 out of the vaccinated group compared to 95 total, and you get to 94 some percent. So those were very close in both groups. The next question was, can you remind me what the next question was?
Oh, sorry. Do we know yet if someone can still transmit the
Okay,
even if they're
yes.
vaccinated?
No, we do not. And that's what is a bit frightening, is that if we're so lucky as to do widespread vaccination all over the world, I think people should definitely continue with social distancing and wearing masks. And the, you really, there's something, some vaccines confer what's called sterilizing immunity, and that means that you cannot get infected whatsoever. If you have this vaccine, you cannot get infected. But other vaccines protect you against developing the disease that's caused by the virus. So one of the poliovirus vaccines is a good example. You can still be infected with poliovirus, but the virus won't spread to your nervous system, so it won't cause paralysis. But you get an infection and nobody would even know they had that infection if you were exposed. So for nobody's had enough time to really look at what's going on since the phase 3 trials, but people who've been vaccinated almost certainly can get reinfected. Now, can they transmit that? Nobody knows. And so, since it's not a vaccine that confers sterilizing immunity, I would think that people should assume that you can transmit it and continue to wear masks and do other things to make sure they're not transmitting the virus.
Got it. So we don't have long-term data from these vaccines yet. So how should we think about the long-term potential side effects? And what have we learned from previous vaccines?
Yeah, that's an interesting question. I think the safety data out there from vaccines that we've all gotten, polio vaccine, measles vaccine, and so on, they're extremely good. And so long term, we have very few substantial side effects. There are some people have severe allergic responses to vaccines, and I just looked up the data. In mid-December, they detected 21 cases of anaphylaxis after administering like 1.9 million doses of the Pfizer vaccine. So it was like, and most of these occurred within 15 minutes of vaccination. So that's a bit more than one person in 100,000 who's vaccinated will have a severe allergic response. But the interesting thing is, in the clinical trials that were done, in the phase 3 clinical trial, they got allergic responses in 0.63% of the people given the vaccine, but 0.5% of the people who got the placebo also had allergic responses. So, there are going to be allergic responses. And so that's why when you get these vaccines, my understanding is you are supposed to wait for 30 minutes and be observed to see if you're going to have one of these responses. If you do, you're given Benadryl or other things that will calm down your immune system.
Got it. So on the topic of side effects, are there any public concerns or misconceptions about these vaccines that you'd like to address? For example, the concern that the vaccine will alter your DNA or cause fertility issues. Can you talk about where these notions might be coming from?
Okay, so the idea that, let's go to the fertility issue first, because I honestly can't understand where that came from. There's a very good piece by Catherine Wu in the New York Times that discussed this. I think it was in December she wrote this. And apparently, I didn't know where this was coming from. This is what I know scientifically. Okay, so a virus, the spike protein on the coronavirus is there to fuse the membrane of the virus to the membrane of the host cell. So lots of viruses have these fusion proteins, okay? And they function in mixing the lipids of each of the membranes and fusing the two biological membranes. So that's the function of a viral spike protein, usually, is in fusion. And then somebody put out a blog that said, oh, well, if you make antibodies against the viral spike protein, there's also a fusion protein that's in the placenta that plays a critical role in developing the placenta. Therefore, it will make you infertile. Well, actually, there is a fusion protein in the placenta. That's true. And there are fusion proteins all over the place. They're involved in many biological functions in our body. The one in the placenta happened to have originally come from an ancient retrovirus that integrated into the host genome. It looks nothing like coronavirus fusion proteins, period. The thing that's in common is the word fusion in the description of its function. And so how this got out is totally beyond me. Someone just read the word fusion and knew nothing about the biology behind viruses or the ordinary fusion events that happen all the time in our bodies. Okay, so that's the first one. The next one is, will the RNA get into your genome? That's the question that's been, that's kind of floating around now too. RNA does not, the RNA, they've done a lot of experiments on this using RNA vaccines, studies over the years. The RNA remains in the cytoplasm of your cell. It will not go into your genome. And there's no evidence for this. And there is, however, a preprint up in a preprint server called bioRxiv. So as people may know, there is a way to post papers before peer review on servers, medRxiv, bioRxiv, chemRxiv, and so on. And so sort of biologically minded people started doing this because it was very successful for the physicists. Okay, so there's a lot. I think these servers are great. Every once in a while, someone posts a paper and it hasn't been peer reviewed that should never be published, and hopefully never will be published in a peer reviewed journal. But a paper did come out within the last month or so saying that under a very artificial situation in which you use cells in culture, you could turn on an enzyme, a natural enzyme that reverse transcribes RNA into DNA. And in that case, you could get the SARS-CoV-2 mRNA into the genome. This is not, we have common cold coronaviruses all the time. They have basically the same machinery to copy their RNA. They do not go into the nucleus. They just don't. And so it's really too bad that this paper was out there, because it was picked up by a lot of people in the news. And then the idea was, okay, if we have a vaccine, it's got RNA in it, the RNA is going to go into your genome and alter your genome, which is a really bizarre thought to have. And I just want to say it doesn't do that. There's no evidence for that. Even if it did, your cell's going to die anyway. So it would make no difference. But anyway, that's not right.
Got it. Yeah, thanks so much for addressing those. And so, although this COVID-19 pandemic is obviously still ongoing, in what ways are you and other scientists preparing for future pandemics?
Yeah, so a lot of people are trying to make a so-called pan-coronavirus vaccine. And so, for example, I can tell you about our work because it's most familiar to me, but uh, we're trying to, Alex Cohen in the lab, a graduate student, is trying to make, well, he has made something that might work as a vaccine that would induce immunity to a wide variety of what are called beta coronaviruses. So SARS, conventional SARS, and SARS-2 are both beta coronaviruses. And then there's a bunch of others that circulate in bats and pangolins and other animals. And so he made a mosaic particle that appears to induce protective, what we think would be protective immunity against a wide variety of these. So there are efforts like that. There are efforts to, for example, try to make what you make is called an immunogen. So try to make an immunogen that would stay in your body for longer than a conventional immunogen. And then through processes in immunological memory, you might make better antibodies if the so-called, if the immunogen is around more, the body has more time to make immune responses to it and perfect those antibodies. And there's some evidence that then becomes more cross-reactive. So I know some scientists are working on that, for example. But basically, the other thing that really is very important is there have been people who go out in the field and they look for what viruses are circulating in animals, in rodents and in bats and so on. And they characterize those, and then they look bioinformatically and so on to predict which ones might be the next pandemic strain. And a lot of this was going on prior to SARS-CoV-2. So the people who were doing that knew this was going to happen. That's what's very frustrating, is if you look at the papers from 2018, 2017, and so on. They all say, here are these viruses. They look kind of like SARS, but they could come in and cause a pandemic. And they were completely right. So that's a hugely important thing that I think it's possible that people don't understand how important it is to survey bat viruses. I mean, before this pandemic, people might have said, who cares what viruses bats get? But it's extremely important to continue to do that.
Yeah. So I just have one more question before we get to audience questions. And it's, again, more on the practical side. So if someone has already had COVID-19, do they still need to get vaccinated?
Well, of course, we don't know the answer to that, but I think they should. And I guess one of the reasons is there are examples where you mount a more potent and longer lasting immune response from a vaccine than you do from the natural viral infection. And a good example of this is the human papillomavirus vaccine, which is given to young people in order to prevent cervical cancer, for example, which is caused by this virus HPV. So the natural immune response to HPV is quite weak and ineffective. The response to the vaccine is much more effective. Now, we don't know yet. We don't have enough data yet to say the immune response to the vaccine appears to be as good, we know that, as the natural infection, but it might, there's a case to be made that might even be better. So I can tell you that personally, if I got a SARS-CoV-2 infection, I would also get vaccinated afterwards.
Got it. Thanks. So now we'll move on to audience questions. Just pull those up. Okay. So we have a question that just came in. What is the evidence or expectation that the COVID-19 vaccine will lead to immunity for people with weak immune systems and older than 75 years old?
That's an excellent question. My understanding of the clinical trials was that they did include older people. Now, I don't actually know if they included people with weak immune systems, immunocompromised people. But for the older people, it appeared that those vaccines worked quite well. So the hope is that it would work. It would certainly, for people who have, it depends on how you're immunocompromised. So there are different mechanisms that would, in some people it might work, in other people's it might not work as well. But I think in everyone it will mount some sort of immune response, and that is more likely to be protective than doing nothing. So I think the bottom line is it's best to get the vaccine. And of course, people who are in this position, they're definitely recommending vaccination for people over 75. They're also recommending it for people who are immunocompromised. And I think anyone in either of those positions should talk to their doctor about this.
Okay, so we have another question. Could getting the vaccine cause a false positive in screening COVID tests, such as the ones being used at Caltech?
If you got the vaccine and then you got the vaccines being used at Caltech right now are nucleic acid based tests. And so I don't know the answer to that, but okay, the question would be, do you have nucleic acid from the vaccine in your nose and when you do a nasal swab? Okay, but actually I just thought of the answer. The vaccine only gives you messenger RNA for the spike protein, and most of the nucleic acid tests look for a protein called N, which is the nucleoprotein. And then they also sometimes look for other viral proteins like ORF1A, I think, and maybe spike. But what would happen is if you got a nucleic acid based test, if you were positive only for the spike, that would immediately say something's wrong here, because there should be more copies of the N gene than there are of the S gene. So the answer would be it should not affect your the type of COVID-19 testing that we are doing. The antigen-based tests, almost all of them, that's where you look for the proteins of the virus rather than the genetic material. Those are against the N protein also, and you're not going to be making viral N protein if you've gotten the vaccine.
Another question, what is the difference between emergency use authorization and regular authorization? And were any testing protocols relaxed or skipped during emergency use authorization?
Yeah, I'm not a government official. I think I want to just leave that to someone who is a lot more legal knowledge than I do. I'm sorry. I could make the obvious answer. One of them's used in an emergency, but I really can't. I don't feel competent to answer that in a knowledgeable way.
Sure, yeah. So when you were talking earlier about the mRNA vaccines and you said, we don't know what's in the mRNA vaccines, were you referring to we as in the general public and people not involved in those companies, or do the companies involved in making the vaccines not fully understand?
Oh, they fully understand it. So there's been like 8 or 10 years of making formulations to package mRNAs. And then, for example, Moderna started to make mRNA vaccines. That company's relatively new. And they've been testing these different formulations for quite a while, several years, including the one that was given in the phase 1 clinical trial in humans against influenza for 2 different influenza vaccines. They know exactly what's in it. And I've seen papers that tell you what are in earlier formations of these lipid nanoparticles. So we know what the sequence of the mRNA is that's encapsulated. We know pretty much what components are in the lipid nanoparticles. We just don't know exactly what's in them because that's proprietary. But you can guess based on the earlier papers. I mean, anyone could go to the literature, find an earlier paper where this was done in animals, and I'm sure it's very similar. And all of these things have been proven to be safe in animal models and humans.
Got it. Okay, another question. We've heard that SARS-CoV-2 has, quote, checking procedures to minimize errors in replication. What does this mean for the variants that are popping up around the world?
That's true. So there's viruses can either have RNA as their genetic material or DNA. And viruses that have DNA generally have a much larger genome. And so they always have proofreading enzymes to make sure they don't make too many mistakes, because there's a certain error threshold at which the virus is totally useless and it doesn't want to exceed that. So now let's go back to the RNA viruses. SARS-CoV-2 and all coronaviruses, 4 of which cause common colds, for example, these are RNA viruses, and their genome is larger. It's 30,000 bases approximately, whereas let's say HIV is about 10,000. So HIV does not have a proofreading activity, but coronaviruses do. And so it's because their genome is much larger than other RNA viruses. They're the only RNA virus with a proofreading activity. So it turns out that if you look at how many mutations there are per replication cycle in a coronavirus, there's like on average 1 mutation for every 30 replication cycles. So every 30 times that it divides and makes copies of itself. And that's like 30 times lower than influenza virus, which is another RNA virus. So the mutation rate is a lot lower. So the suggestion would be that the virus that passes on from one person to another has like 1 mutation, let's say, or maybe even none. So that's a pretty slow mutation rate. I don't know what this means, the variants that they've detected. They have spike mutations, but as I said, they have mutations in other viral genes. And people are frantically now doing experiments to figure out what all those mutations mean. Some of them are going to be just random. They will have no effect on anything. But why are there so many? And that is something that bioinformaticians will have to, we're going to have to answer that, we meaning the whole community. And my lab doesn't do any of this type of sequencing, but there's going to have to be a lot more sequencing to answer that question. And the reason this was detected was the UK does a lot of virus sequencing. The US has been doing less. So it's hard to kind of trace what happened in the US. Like, did you have variants that had like 2 or 3 of those mutations, and then somehow those changed to have 4 or 5 of them, and then up to the number they have, which is something around 17, I believe, changes from the original strain that went into the vaccine, let's say. So I think it's too early to answer that, but it is something that really bears looking at. There are methods in coronaviruses to recombine. So let's say you were infected by 2 different coronaviruses, you might recombine your messenger or your RNA. And so maybe that would cause a buildup of mutations faster, but that just vast amounts of sequencing and analysis needs to be done to answer that.
Yeah. Are there different mechanisms for how mRNA will be transcribing proteins in different individuals to produce antibodies, given that everyone's DNA is different?
Well, mRNAs are translated in the cytoplasm into proteins. And so this involves a common mechanism where the messenger RNA is read on ribosomes in the cytoplasm now. And then transfer RNAs carry amino acids according to the genetic code. So every 3 bases of RNA will encode a particular amino acid. And so there's a lot of like really beautiful YouTube videos about protein translation in the cytoplasm. It's basically the same in all humans. It's the same in mammals. It's not dependent on your DNA unless it's in an incredibly subtle way. But this is conserved. This is why you can study protein translation in, a popular system to study it used to be dog pancreatic microsomes. So they would get extracts from dog pancreas, and what they learned about translation was the same as what happens in humans.
Okay. Another question. Could you discuss how the AstraZeneca DNA-based COVID-19 vaccine works compared to the BioNTech Pfizer mRNA vaccines?
Yeah, okay. So there's 2 of these vaccines. One is the AstraZeneca and another is the Janssen or J&J vaccine. And they're both, what they do is they use another virus as a scaffold, and they're both using adenoviruses. So adenoviruses give us some forms of common colds, but what they've done is they've gutted the entire virus using molecular biology techniques, and then they replace it so that it can only get into a cell, and after that it can't make new copies of itself. So it's replication incompetent, but it carries the gene now in the form of DNA for the spike. And so what happens is, instead of delivering an mRNA, which is translated in the cytoplasm into the spike protein, which then goes to the surface of that cell, they use a vector, which is a gutted adenovirus that will bind to a cell, get its DNA into that cell. That DNA will go to the nucleus of the cell and be transcribed into RNA, and then the RNA will get out into the cytoplasm, and then it's the same thing as the mRNA vaccines. So it's another way to deliver the genetic material encoding the spike. And the difference between AstraZeneca and the J&J vaccine is they use 2 different types of adenovirus. So AstraZeneca uses a chimp adenovirus that's been completely gutted. It can't hurt you. And you also wouldn't have been exposed to it, so you wouldn't mount an immune response against it. And J&J uses an adenovirus called Ad26, which people can get infected with, but for, again, unknown reasons, people just don't mount much of an immune response to it, so that you could get infected with it from the research people have done over and over again. So you could use this Ad26 vaccine, the hope is, based vaccine numerous times. So it's a different way. The short answer is it's a different way of delivering the genetic code for the spike protein.
Okay. What do you see the future of vaccine development looking like? Are there any particularly exciting technologies that will change how we do things? And I think we just have time for just this last question.
Okay. Well, I think that that's a really short answer. I think that the incredible efficacy of the mRNA vaccines has been such a wonderful surprise and relief to everyone. I expect that a lot of vaccine formats will convert to this mRNA technology.
Okay. Well, thank you so much, Professor Bjorkman, and thank you to everyone for joining us. Be sure to sign up for the next Conversations on COVID-19 webinar on February 3rd, where my colleague Robert Perkins will talk to Professor Chiara D'Arraio about wearable medical technology for temperature sensing. And in the meantime, if you're interested in exploring more of the science behind viruses and COVID-19, please check out the Caltech Science Exchange, which launched last spring to provide trustworthy answers, clear explanations, and fact-driven conversation on critical topics in science and technology. and you can find it at scienceexchange.caltech.edu.